Thank you, Ministers and Chief Medical Officer, Dr. Parasram, and good morning to members of the media and to the viewing and listening public. There's been a lot of discussion recently in the media about the immune response to COVID-19, the COVID-19 virus, which is SARS-CoV-2, and in particular about people who recover from the SARS-CoV-2 infection and the question of whether they're protected against reinfection, and if so, how long does that protection last? Now, despite all the recent headlines, the short answer to that is that no one knows. Scientists are still trying to determine exactly how COVID-19 immunity works. There are clues, but the picture is far from complete. Now, before I go into what is currently known, I first want to remind you that our immune systems use several tools to fight viral infections, and each type of virus requires a slightly different set of tools. Now, these tools include special proteins called antibodies, which recognize and bind to viral proteins. Some antibodies work by binding to the virus particles in such a way that they prevent the virus from infecting and replicating in their target cells. And these antibodies are called neutralizing antibodies. Now, the immune system toolkit also includes special immune cells called T cells that recognize and kill cells that have been infected so that they can't produce any more virus. Now, the first time the immune system encounters a particular virus, it can take a few days for it to size up this invader and then to make and use the right set of tools to clear the infection. And after an infection with a given virus, the immune system remembers what it learned about how to protect the body against that specific virus. So if it encounters that same virus again, it can mount a defense much more quickly and prevent the infection from taking hold, or at least prevent it from causing illness or from spreading to other people. This is what we mean when we say someone has developed immunity to a particular virus. Now, for some viruses, immunity can last a lifetime, while for others, it may only last a few months or years, after which time reinfection is possible. Often with the second infection being less severe than the first, although this is not always the case. There are also some viruses that the immune system never manages to clear completely. Now, the first clue to where SARS-CoV-2 immunity might fall on that spectrum came from other coronaviruses known to infect humans. Infection with these other coronaviruses results in the production of protective antibodies, we know that, and the level of protection depends on the type of virus and also the severity of infection. So you produce these antibodies, but with these other coronaviruses, those antibodies typically decline. So they go down to undetectable levels within about 3 months to 3 years. And that depends on the type of coronavirus and the severity of the infection the person had. Occasionally, it lasts for longer. Now, in general, the more severe the infection, the stronger and longer lasting the immune response. This is what we know about the other coronaviruses that are out there, other human coronaviruses that cause like common cold and stuff. Now, for some of these other coronaviruses, studies showed that initially, when antibody levels are high, there is protection against reinfection. But as the antibody levels fall, this so-called sterilizing immunity is lost and reinfection can occur. Based on these data, scientists hypothesized that antibodies to SARS-CoV-2 may provide protection for at least some period of time. We now have strong evidence from multiple studies that most people develop antibodies against SARS-CoV-2, and they do that by about 1 or 2 weeks after infection, and that the antibodies produced include strong neutralizing antibodies that remain at high levels for a few weeks after infection, and then they typically begin to decline, so similar to the other known human coronaviruses. A recent study out of the UK that has been widely reported in the media found that on average, neutralizing antibodies were first detected about 2 weeks after the onset of COVID-19 symptoms. They rose to a peak level by about day 23, and then there was a rapid decline. As in previous studies, they also found that people with less severe disease had lower peak neutralizing antibody levels than those with more severe disease. Also, they found that in the people with the lower antibody levels, the neutralizing antibodies dropped to undetectable levels in just under 2 months, so they didn't last very long. People who started off with higher neutralizing antibody levels also showed a significant decline over time, but their antibodies remained detectable at the end of the 3-month study period. So further follow-up is needed to determine if their antibodies will also eventually become undetectable. Now, many news outlets reported this study. They reported that this study confirmed that SARS-CoV-2 immunity is short-lived and that people can be reinfected within months of recovery. In fact, what the study showed, and which is also important to note that this study hasn't yet been peer-reviewed, what the study showed is that in some people, neutralizing antibodies are short-lived. Now, in light of this, and based on what we know about the other coronaviruses, the authors of that study, of the paper describing that study, they suggested, but they certainly did not confirm, that reinfection might be possible in these people. However, the authors also noted that, and as I also indicated earlier, the immune system is comprised of much more than just neutralizing antibodies. So measuring neutralizing antibodies can give you clues as to what might happen, but it cannot tell the whole story. For example, we don't know whether the immune T cells play an important role in providing protection against reinfection or reducing symptoms. And even if the neutralizing antibodies that they measured are a critical component, we don't know what levels are required to confer protection. Animal studies suggest that even though low levels of SARS-CoV-2 neutralizing antibodies might not protect against reinfection, they may be sufficient to prevent disease symptoms. Also, research on other coronaviruses where reinfection was shown to be possible, they noted that reinfected individuals did not experience any symptoms. So in summary, any conclusions about reinfection with SARS-CoV-2 based on antibody studies is premature. And we can, however, we can't make any firm conclusions and say, yes, people can be reinfected, or no, they can't be reinfected. But we can ask the question of whether there have been any confirmed cases of reinfection, and so far the answer is no. Now, there have been sporadic reports around the world of people who tested negative. They had COVID-19, they were in recovery, they tested negative by PCR twice, and were therefore considered cured. And then as many as 1-2 months later, they tested positive again. And this led to the suggestion that people were being reinfected. However, the PCR test, which detects the presence of genetic material from the virus, cannot tell whether the viral genetic material that is detecting is due to the presence of infectious virus particles or if it is just non-infectious viral debris. The only way to confirm this is by taking a sample from the patient and trying to grow it and trying to actually grow the virus to see if the infectious virus is still there. And so far, in cases where this has been done, the results indicate that there is no infectious virus, and what is being detected, months after somebody has recovered and looks like a reinfection, what is actually being detected is this viral debris. This also appears to be the case in people who consistently test positive weeks after their symptoms have disappeared. However, since the routinely used tests cannot confirm one way or the other what it is, if it's infectious or non-infectious, the protocol being used is to keep testing until there are 2 negatives. Finally, there are some people who oscillate back and forth between negative and positive PCR results. Now, all tests have a limit of detection, below which they won't detect their target even if it is present, resulting in a false negative result. Now, PCR tests are extremely sensitive, but if the level of virus in a patient sample is below the test threshold of detection, it will return a false negative result. Now, false negatives are most likely early on in an infection when the virus is only just beginning to replicate, so the levels are very low, and again, late in an infection after the immune system has cleared the majority of the virus. During these times, very slight variations in virus levels or in test conditions can mean that on one day you don't detect the virus and the next day when you retest, you detect it. Again, this is why They wait until there are 2 consecutive negative tests before someone is discharged. Thank you very much.